What if I told you that your next biologics process could run continuously for 30 days straight, with real-time monitoring catching every deviation before it impacts product quality? That your 5,000-liter fed batch bioreactor could be replaced by a 500-liter continuous system with higher output, and that your downstream bottlenecks could virtually disappear? For many bioprocess scientists, this sounds too good to be true. Yet, it's already happening. In fact, in industries like automotive manufacturing, it has been standard practice for many decades. In 1913, Henry Ford revolutionized manufacturing with a continuous assembly line that reduced the time to build a car from 12 hours to just 90 minutes. More than a century later, most biologics manufacturing still operates like pre-Ford factories, batch by batch, with starts and stops, idle equipment, and inefficiencies that we've simply accepted as the cost of doing business. But what if we're at our Ford moment in bioprocessing? Welcome to the Smart Biotech Scientist, and I'm your host, David Brühlmann. Today, we're exploring how continuous manufacturing is transforming biologics production from an art of batches to a science of flow. The numbers tell a compelling story. Up to 30% lower production costs, facility footprints reduced by half, process intensification delivering 10 times higher cell density, and time to market accelerated by months. And what's the most exciting part? This isn't happening just at pharma giants with billion-dollar budgets. Small and mid-sized biotech companies are implementing continuous processes and gaining competitive advantages their fed batch-focused competitors can't match. If you're a process development scientist, a C-level leader, or a biotech founder trying to do more with less, today's episode will challenge everything you thought you knew about biologics production. Because as I often tell my clients, science may give you headaches, but biologics manufacturing doesn't have to. The continuous revolution is here, and those who embrace it will leave the fed batch bound competition behind. Let me paint a picture familiar to many of you. It's Monday morning and you're reviewing timelines for your company's lead candidate. Your CEO wants to accelerate development while cutting costs. Does it sound familiar? Oh, you bet. Your manufacturing team is concerned about facility capacity, and regulatory is asking about process consistency and control strategy. This is where continuous manufacturing enters the picture. It's not just a buzzword, it's a paradigm shift that could be the solution to many of the challenges keeping you up at night. The continuous bioprocessing market is experiencing remarkable growth, valued at approximately $218 million in 2023 and projected to reach $599 million by 2028. That's a compounded annual growth rate of 22.4%. But what does this mean for you, the scientist in the trenches of development? Recent industry surveys reveal some fascinating trends. According to Bioplan Associates, the 20th annual report and survey of biopharmaceutical manufacturing, over 68% of facilities are likely to adopt a continuous chromatography system at some scale in the next 2 years. Even more striking, 72% expect to see adoption of upstream perfusion in most facilities. Let's break down why continuous manufacturing matters for your daily work. You will see increased productivity and volumetric efficiency. Your facility maintains constant production without the non-productive steps of setup, takedown, and cleaning between the batches. You will see improved product quality and consistency, which means a more uniform condition that will lead to more consistent product attributes, and you will also see a reduced facility footprint. As we'll see in one of the case studies coming up, 4,000-liter single-use bioreactors in perfusion mode match the output of 4 12,500-liter stainless steel fed batch bioreactors. And finally, you will see enhanced process control, because real-time monitoring allows for immediate adjustments. Now, you might be wondering, how do different companies actually approach continuous manufacturing? Well, as our industry moves away from traditional fed batch processing, I've seen 3 distinct strategies emerge. And trust me, understanding these approaches will help you make informed decisions for your own facility. Let me break them down for you. Number 1, perfusion cell culture for upstream processing. The second is continuous downstream processing. And then the third one is the integrated continuous bioprocessing, so the complete end-to-end solution. But before we get ahead of ourselves and reveal all our cards, let's examine a few compelling case studies. My recent experience perfectly illustrates this shift. I had the opportunity to visit the development center of a large biotech company, and what I heard there was eye-opening. Their leadership team made a bold declaration. All future molecules will be produced using continuous manufacturing, both upstream perfusion and continuous downstream processing. What struck me most was their certainty. They weren't hedging their bets or keeping traditional fed batch as a backup. They simply didn't see any compelling reason to maintain fed batch platforms for their new molecules. This isn't just another case of a company testing the waters with continuous manufacturing. This is a major player in our industry making a definitive statement about the future of bioprocessing. And they are not alone. Let's look at the prime example of continuous manufacturing's transformative potential in the industry. Amgen's $200 million next-generation biomanufacturing facility in Singapore represents one of the most ambitious implementations of continuous bioprocessing to date. The facility incorporates cutting-edge technology, including flexible single-use bioreactors, disposable container systems, integrated continuous purification processes, and advanced real-time analytics. What makes this facility particularly noteworthy is the dramatic efficiency gains they have achieved through continuous manufacturing principles. The numbers are staggering. First, an 80% reduction in facility footprint compared to traditional fed batch manufacturing plants, leading to significantly smaller environmental impact and reduced construction costs. Second, unprecedented speed of deployment. The plant was built in half the time of conventional facilities while requiring less capital investment, demonstrating how continuous manufacturing accelerates production capacity. Third, and perhaps most compelling for your finance team, the reduction in operating costs is significant. Amgen projected cost savings of 60% or more per gram of protein. These savings come from multiple factors. Number 1, increased resin utilization, then from reduced buffer consumptions, lower labor requirements, and less energy consumption. The facility uses single-use bioreactors, disposable plastic containers, real-time quality analysis, and modular design. These are all key elements that support continuous manufacturing. According to Amgen's own statements, these innovations are expected to result in a cost reduction of 60% or more per gram of protein. This facility demonstrates how continuous manufacturing isn't just a theoretical concept. It's being implemented at commercial scale by industry leaders who recognize its tremendous potential for improving efficiency, reducing costs, and maintaining high product quality. You likely have heard of another example in continuous manufacturing. You likely have heard of Sanofi's Factory of the Future in Massachusetts that was launched in 2019. This highly digitized, paperless manufacturing facility implemented integrated continuous bioprocessing using perfusion-based cell culture with single-use technologies. The facility achieved remarkable environmental benefits, a 94% reduction in chemical usage, 80% less energy consumption and CO2 emissions, 91% water savings, and 321 tons annual waste reduction. The design optimizes for flexible operations and rapid production changeovers, maintaining 99% facility uptime. Another compelling case study comes from KTH Royal Institute of Technology in Sweden in collaboration with AstraZeneca. They developed a small-scale perfusion system for HEK293 cells producing recombinant human EPO. The team developed a novel small-scale perfusion bioreactor system using commercially available benchtop equipment modified specifically to accommodate the more sensitive HEK293 cells. Their approach enabled higher oxygen transfer under milder conditions that would be needed for the typical CHO cells. The results were remarkable in three key areas. Number one, they maintained HEK293 cell densities at or above 80 million cells per mil, approximately 10 times higher than typical fed batch processes, using low cell-specific perfusion rates of just 10 picoliter per cell per day with enriched feed medium. Second, they demonstrated extraordinary process longevity by maintaining the culture for more than 75 days. Third, and perhaps most significant, they achieved these results with human cell lines which produce proteins with more human-like post-translational modifications than the CHO cells that dominate our industry. This study demonstrates that continuous manufacturing technologies can be implemented effectively at small scale and with human cell lines. The case studies we've just seen demonstrate the remarkable potential of continuous manufacturing across different scales and applications. It can deliver substantial improvements in productivity, efficiency, and quality. Now that we understand the landscape, let's discuss how to actually implement continuous manufacturing in your facility. What are the key technologies and approaches you should consider? First, let's talk about process analytical technology, or PAT. PAT serves as the cornerstone for continuous bioprocessing by enabling several things. It enables real-time monitoring of critical process parameters. It enables the implementation of feedback control loops, also quality by design implementation, and it enables a cost reduction and waste minimization. The second approach focuses on the purification. Continuous chromatography systems are critical for downstream processing, and they will, as we'll see, substantially enable to save costs, especially for protein A chromatography. The third one encompasses everything with respect to cell retention. So we see cell retention devices such as tangential flow filtration, so the TFF, or alternating tangential flow ATF systems, and they are essential for maintaining high cell densities in perfusion cultures. Recent technological advances are making continuous manufacturing more accessible. For instance, single-use technologies provide flexibility for continuous setups. Automation and digital transformation creates robust process control, and advanced downstream innovations, including continuous ultrafiltration and diafiltration and continuous chromatography, make those steps much more efficient. However, it's important to address the elephant in the room: the challenges of implementation. According to Bioplan Associates' latest industry survey, despite high interest, there's a significant gap between aspiration and practical adoption. While 72% of respondents expect to see widespread adoption of perfusion in the future, many identified specific barriers. 55% believe continuous upstream processing is too costly. Over 50% find continuous upstream processing too complex. And 62% consider continuous downstream processing too early to adopt. I would love to hear what you think about continuous processing. One way you can do that is by leaving a review on the platform you found us on. I'm looking forward to hearing from you and to seeing actually what you think about continuous processing. There is also a talent gap. Finding, training, and retaining staff with expertise in continuous bioprocessing remains a significant challenge. In fact, process development roles in both upstream and downstream ranked among the most difficult positions to fill. And one misconception worth addressing is that continuous manufacturing requires an all-or-nothing approach. In reality, many companies are implementing partial continuous processes or hybrid approaches, allowing for gradual adoption rather than a complete transformation. Now, let's examine a fascinating real-world downstream implementation described by researchers at Paul Life Sciences by Joka, I hope I pronounced this name correctly, and Al, who developed a continuous chromatography platform for monoclonal antibody purification. What makes this case study particularly valuable is the quantifiable impact they achieved. The team successfully implemented a multi-column chromatography process that delivered 3 remarkable improvements. Let me walk you through the setup they used. The team integrated 4 critical unit operations across 2 Cadence Bio SMB PD multi-column chromatography systems. They had a protein A capture, a viral inactivation, a flow-through anion exchange, and a mixed-mode cation exchange chromatography. Here is what made this implementation particularly impressive. They managed to process 25 liters of harvested cell culture fluid in less than 12 hours. That's the kind of efficiency that makes continuous processing so compelling for modern biotech operations. Now let's look at the improvements. First, they increased productivity of the protein A step from 13 to 50 grams per liter per hour, and of the mixed mode cation exchange step from 10 to 60 grams per liter per hour. This dramatic improvement came from optimizing load conditions and continuously cycling their chromatography columns. Second, they reduced buffer consumptions by 44% compared to batch processing. This addresses one of the most significant operational costs in bioprocessing: the preparation, storage, and disposal of process buffers. Third, they reduced resin volume required for protein A and mixed-mode cation exchange chromatography by more than 95% compared to batch, while the anion exchange was reduced by 74%. While these benefits of continuous manufacturing are compelling, successful implementation depends on key technological enablers and strategic planning. Your organization needs to leverage both cutting-edge technology and careful assessment of current capabilities to create an effective adoption roadmap. When you're evaluating your facility's readiness for this transformation, focus on these critical enablers and success factors. Number 1, advanced bioreactor systems and cell retention devices for upstream operations. Then focus on continuous chromatography and single-pass filtration systems for downstream processes. Third, process integration capabilities and robust control systems. The fourth step is data analytics infrastructure for real-time monitoring. And finally, the fifth is the team expertise in continuous bioprocessing operations. And here is a key insight many overlook. Implementation should be driven by technology readiness and operational maturity. The most successful transitions often begin by implementing continuous processing in specific unit operations where enabling technologies are well established and can deliver immediate impact. This systematic approach allows you to build expertise, validate technology solutions, and demonstrate ROI while minimizing operational disruption. We have covered a lot of ground today in our exploration of continuous manufacturing for biologics. From Amgen's revolutionary facility in Singapore achieving 80% footprint reduction to KTH's remarkable achievement with HEC-293 cells at densities 10 times higher than traditional methods, we're seeing the continuous revolution unfold before our eyes. The technology enablers we discussed, from advanced cell retention devices to multi-column chromatography systems, are making continuous processing more accessible than ever before. And case studies like PA Life Sciences implementation demonstrate the dramatic improvements in productivity, buffer consumption, and resin utilization that are possible today. However, as the Bioplan Associate survey revealed, significant implementation challenges remain. Concerns about cost, complexity, and technical expertise continue to create a gap between industry interest and widespread adoption. In our next episode, we'll tackle the remaining critical questions on your mind. What do regulators think about continuous manufacturing? And what's the real economic impact? We'll explore the evolving regulatory landscape, analyze detailed cost comparisons, and share additional case studies that demonstrate successful implementations across various scales. If you're intrigued by the technical possibilities we've discussed today, but need to understand the business case and regulatory pathway, you won't want to miss part 2 of our continuous manufacturing deep dive. And if you have any questions about continuous process development or need guidance on implementing these strategies in your facility, I'm offering you a free consultation. Book your session at Brühlmann Consulting/call. That's Brühlmann Consulting/call. Thank you for joining me today. I'm David Brühlmann, your host, and I look forward to seeing you in the next episode, where we will continue our exploration of continuous manufacturing. All right, smart scientists. That's all for today on the Smart Biotech Scientist Podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.